right? If we're talking about right? If this is the mRNA, we always set up our reading frame, or how we determine which parts are a codon, by the first AUG closest to the 5' end. Yes? A constructive? No idea. Was it a wrong answer? Yeah, it was something made up. When you run out of real things for wrong answers, you just make shit up. The promoter is where the sigma factor and the RNA polymerase binds, right? Don't get mixed up, right, translation, transcription. Yeah, the operator is where the repressor binds and physically blocks access of polymerase to get through. The operon is the whole thing, promoter, operator, gene. Like, yeah. So the operon in prokaryotic gene regulation is how a regulatory unit might be set up. Right? Whereas in eukaryotes, a regulatory unit is promoter gene. That's pretty much all it is. There's no operator, right? There's never a promoter gene in eukaryotes, right? We have to do them all separately. But in prokaryotes, sometimes we transcribe 3 or 4 genes all together into one big RNA and then just translate them into 4 different proteins. Yeah, the inducer is generally anything in the prokaryotic operon that causes ultimately transcription to turn on, induces that process to go. In the case of lac operon, that's lactose, because it binds the repressor, removes it from the operator. Now RNA polymerase can just go. There's nothing sitting in its way to stop it. Exactly. And so that's negative regulation. If something bound to hold it off, that's a negatively regulated operon. That cool? We cool with that? We like it? We dig it? It's our friend? No. Okay. All right, so let's go now. So that was all transcription, regulation. Back to translation, right? And so if we have, right, we always start at the first AUG. That sets up our reading frame, and that's what we call how the ribosome goes through. So again, that's kind of like, if there's a question about how do we decipher, how do we figure out, how do we control the sequence of a protein, it's based on the reading frame of the mRNA, of how the ribosome reads it. It's not the triplet sequence, it's not the genetic code decipheron, it's not some other made-up term, it's the reading frame. And that's important because if we screwed that up and we did, let's forget that we have to start at the first AUG, and I just start where Malone starts writing nucleotides, now CCA codes for something completely different than AUG. You're not going to get a methionine there. Right? And then you're going to do this, and then you're going to do this, and then you're going to do this. You're not going to hit a stop codon. It's going to be all different amino acids in that protein. It's not going to be the protein you're trying to make. Right? So if you get questions where you are given a 5' end, blah, 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 and I say translate this, and you start here, you're going to, if it's a written question, you're wrong. And if it's a multiple choice, I'm going to have the answer to this as one of your options. Right? Because you know how I am. So that way you can't just go, oh, that didn't show up in the multiple choice, so, don't forget, you have to set up your triplet correctly at the first AUG. That will be a viable option for you. You have to know how to do translation of an mRNA, okay? And that's the reading frame, right? You can have, I guess I should give an example of that. So if I say 5', so I don't have to keep writing, right? Again, if you start from down here, do that. Okay? We want to start with the first AUG closest to the 5' end. Right? So we'd want to start our reading frame here. That would be our first amino acid. We know that's methionine. And then after that, it's whatever, using your genetic code chart to figure out what they are. Until you come to a stop codon in frame. Right? It doesn't matter if... I don't know what I'm doing here. If there's a UGA right there, right? Is that a stop codon? No. That's a sequence of UGA that happens to be in sequence 5' to 3', but it's not in the reading frame, so it's not a stop codon. Right? The reading frame is really what sits in that A site of translation. If the whole UGA is not in that site, the release factor can't recognize it. Right? CAU is in the A site. tRNA, complementary, that's going to sit there and kick off its amino acid. The next one is GAC, tRNA is going to come in there, that's anticodon is complementary, that's the next amino acid. Right? It's not a stop codon if it doesn't fall into this frame, this triplet reading frame. Yeah.
Just out of curiosity, when you're translating the untranslated region or the 5' thing before the AUG, you translate it from 5' to 3', correct? So it'd be like CCU?
We don't translate that at all. Right? We transcribed it. Right? Because really, this was the +1 of transcription, right? But that does not get translated because we don't start till here. It's a great question. So this is the +1 of translation. Hang on. Yeah. Well, if you go through the process, if you're on the template strand and it's CCC, is that what it said? Yeah, the template strand CCC, which means you transcribe it to GGG, which means the anticodon would be CCC. Yeah. So you can always actually go through the process of transcription and understanding that to not need to memorize, okay, the tRNA is the same as the template strand. Right? You could memorize that, or you could just do your process and come up with the answer because you know all the rules. Yeah. Well, if it's T's, then you're going to have A's, and then it would be U's in your anticodon. Exactly. But yeah, you can always step through the process instead of just, is it non-template? Is it template? Which one looks like the anticodon? Right? You can always just step through it without having to, have it in your head. Yes, this is how it is, and then check your answer by doing the actual transcription from the template, and then how the anticodon. Yeah, sorry. Yeah, so if we're talking uke, so this I was cheating and just doing probes, because that is actually the 5', her question was, where's the 5' cap? So here's really the 5' phosphate sticking off of that guy. That's the first nucleotide of our mRNA. And then we'd have, draw it like a circle, right? Our methylated guanine, that 5' weirdo bond over there, exactly. So if it's a uke, and then the end, we got to have our tail on it, on the 3' end if it was a uke. Okay? Exactly. Right? So yeah, we never, ever, ever translate that, because if you think about what does this mean, untranslated region. Right? Not translated. That's what UTR stands for. So also, so then when you're on the, right, exactly, when you're on the exam and you're like, holy shit, I can't remember where to start it, they start codon is the name, okay, well, right, start translation there. But still, it is, if it's brand new, it's still easy to go, I can't leave all these nucleotides there, they're just sitting there doing nothing, right? And then remember, UTR stands for untranslated, not translated. So those guys just sit there, okay? And in prokaryotes, which we're just about to talk about, within this sequence, which I didn't draw because that's, I'd have to be drawing a million nucleotides, is a sequence of nucleotides that's called the Shine-Delgarno sequence. Okay, and it's usually pretty close to the first AUG, but it's within the 5' UTR, and that's where the small ribosomal subunit grabs it. That's how it recognizes where it needs to sit to set up translation. Okay? So if we were being crazy and I said, please draw me an mRNA, and you guys were losing your minds, you had a moment, and you drew me an mRNA like this, and first of all, you'd immediately know that I would be jumping up and down and having a cow. So let's just say you were just panicking and you had a lapse. And you started your mRNA with an AUG. And we're talking prokes. Remember we said we never start an mRNA with AUG, ever, ever. No. Right? So let's just say you had a moment, you forgot about my conniption fits, and you were thinking through the process rather than trying to memorize, and you thought, okay, I know the start codon starts translation. What is the process? The small ribosomal subunit has to grab the Shine-Delgarno sequence in the 5' UTR to start. Oh shit, I don't have a 5' UTR. So translation cannot happen. Right? So then you go, oh my God, and you laugh. She didn't get me on that. We're not going to memorize Shine-Delgarno. We're just going to pretend like it's in there. Right? And now you've got this 5' UTR with this sequence in it, which we're not going to memorize. And again, it's just a sequence that the small subunit recognizes. The small subunit is RNA. It likely recognizes it by what? Complementary base pairing, RNA to RNA, anti-parallel. Right? The small ribosomal subunits are RNA and protein. How does it know where to sit? Hydrogen bonding in chemistry. No thinking, right? Bumps into your RNA and hydrogen bonds in a region where the pocket of that small subunit is open. RNA is single-stranded. It's not already hydrogen bound to something else in its tertiary structure. Hydrogen bonds to the Shine-Delgarno sequence, which makes its butt big enough to overlap the first AUG. So now there's a P site there. Ooh, tRNA with methionine can sit down by complementary base pairing of the tRNA to that codon AUG, and then the miracle of life continues. Right? Translation. Okay? So you can always check yourself thinking through these processes as long as you know how they work. You can't forget the 5' UTR if you know that's where the small subunit has to bind to set it up. And then we'd set up our reading frame, and then this would be our stop. And remember, there's no tRNA. It's a release factor protein that then causes explosion of the subunits of the ribosome, and the ribosome falls off at a loss for what to do, and the peptide is free, and then primary, secondary, tertiary, quaternary structure will happen if that's the protein for its final function. Right, the mRNA is always 5' to 3'. Yeah, not the tRNA. So so this one, right, each one has a 5' and 3' end, so the AUG is 5' to 3', right, all of those, and then, yeah, the has to be 3' to 5' on the tRNA. Well, there's my tRNA. We're always reading down the mRNA 5' to 3' in the 3' direction. So it follows our same rules for replication, transcription, translation. It goes down, synthesizing in the 3' direction. When we actually talk about the protein, right, remember we talk about N-terminus, C-terminus, right? That's the polarity of the protein. But yeah, as far as, right, the ribosome moves this way down the RNA, so in the 3' direction, towards the 3' end. So the reading frame is 5' to 3'. Yeah.
The prokaryotes with 5' UTR is called Schindel-Garno.
Part of within the 5' UTR, there is a sequence called Schindel-Garno.
Okay.
Yeah.
What about in Ukes?
In Ukes, it's actually the small subunit recognizes the cap and then slides across the 5' UTR to get to the first AUG. So again, if the cap were right on the AUG, the small subunit is big enough that the P site wouldn't be at the AUG, it would be somewhere else. So there has to be 5' UTR to get the cap far enough away so that when it binds to slide, it'll get right to the, it won't pass up the AUG. Okay, so here's large subunit, small subunit. Ribosomes are very, very similar in structure and function to Ukes and Prokes. The Ukes are just bigger, so they call this 50S and 30S, and Ukes, I think it's 60 and 40. They're just bigger, and that's a unit of sedimentation, blah, 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 which we don't care about. But essentially the same, okay? Same function, almost between the two, conserved evolutionarily. Okay. When we talk about the actual structure and how it sits, we watched that stupid little video, and that video ignored the E site, which is fine, and it kind of depends on what textbook you're looking at and who's writing it and how they think it's easiest to understand. The E site really is just where a naked tRNA, the one that's already lost its amino acid, just falls out. It exits. So the E site is really the exit site. So there's plenty of books and they just say it falls out of P. So when it shifts, it's technically an E and then it immediately falls out. Okay, so in the video we only had A and P. In the book and all of these they do A, P, and E. And it is a little easier to, then every time it shifts, everybody has a spot. Okay, we'll go through the shifting and I'll point that out. But E is just where it falls out. P is for the peptide, because that's where most of them are holding on. A is the acceptor site. Aminoacyl tRNA site, that just means an amino acid attached to the tRNA can go in there, right, the next one. And so we're going to go through the process, but this is just sort of the setup, since it was different than that little video. Okay. So essentially two major catalytic events need to happen during translation. Okay, we need to make a bond, make the peptide bond between the amino acids of the existing one on P and the new one that came in A, right? So that's the condensation reaction catalyzed by the large ribosomal subunit. That's the guy that makes the peptide bond. But we also, so that's this one. But we also have to break a bond, and the breakage happens in the P site. We have to break the covalent bond between the amino acid and the tRNA. Because if we remember that picture from last time with, let's see if we can find it really quickly. If I have the power to just peek at it. There we go. Come on. You can show it. Right? This amino acid serine is covalently attached to the 3' end. We don't have a name for it. Well, there is a name, but we don't need to know it. And we don't even talk about that, how that happens, how it attaches. We break that bond, and the large ribosomal subunit has that enzymatic activity to break it off. So this bond is broken from the 3' end of the tRNA and the amino acid after the amino acid is, or at the same time as it's peptide bonded to the guy in A. So let's go back and look at that picture. Right, so this bond here gets broken while we make the bond between the two amino acids. Sorry. Oh, it's not going to let me erase it. Okay. Okay, so those two things. So we break a bond, make a bond, every time there's like a snapshot of translation. We don't really talk about it in here, and I don't know the exact biochemistry of this bond interaction at that point, but my best guess, as far as what evolutionarily would make sense, is the breaking of that bond releases energy to help make the next. But I don't know if it's enough that it's a complete wash or if there's extra ATP that we need either. I'd have to go look in biochem. Because in replication and in transcription, that's a real high energy bond that we're breaking. The phosphates off of the new nucleotide to put in. Right? That's essentially ATP, CTP, GT, like that's high energy. That's enough to catalyze, that reaction. I don't know if it's enough, just that breaking to make, or if we need, I'm guessing we need extra, but I don't know. I'd have to look it up in biochem. I haven't thought about that in a long time. But yeah, so, and in 380 we do a little more. 360 we don't really even talk about the energy. We talk more about what happens to all the molecules. But yeah, it makes sense because you have to break that one. You're going to release some energy. Why? And it's right there, in that same spot. Why we wouldn't funnel that right into making the next bond? Coupled reactions are pretty much how life exists. But I'm guessing we probably need some extra too. Okay, so let's walk through this process, right, from just the slide. And so, like we said, the ribosome binding site, right, is known as the Shine-Delgarno sequence. Okay, so a lot of textbooks are trying not to use that term anymore, and they're calling it ribosome binding site. But there's still plenty of standardized tests and plenty of other textbooks that use Shine-Delgarno sequence. Again, Dr. Shine and Dr. Delgarno are the ones that recognized the sequence and did all the experiments to figure out that's where the small subunit docks. So they obviously named it after themselves. So it is the ribosome binding site. That's better for students to remember because the name of it is what it does. But it's also classically known as the Shine-Delgarno sequence. And it's within the 5' UTR, right? If we look, we zoom in. Ooh, notice how we zoomed. Right here, right? If this is the start codon, this sets up our reading frame. All of this is 5' UTR. So we have to have that. We're not memorizing the sequence, but it is the same in all mRNAs of prokaryotes. Okay? We're not memorizing the sequence. That's just dumb. Okay, but that's what, right? This guy, the small subunit, grabs it by that sequence. Right, sets it up so this is the P site. Right, this would be the E site and this would be the A site. So it's APE spelled backwards. Or the Environmental Protection Agency. Okay. Whatever. I like APE. Going the wrong way better. But, and so without that sequence, the small subunit couldn't grab it, we get no translation. You have to have that 5' UTR. And if we mutated the shit out of Shine-Delgarno and made it a whole bunch of C's, translation's not going to happen either. Okay, so that sequence is really important and its location. If we put this sequence someplace else in this mRNA down here near an AUG, small subunit would grab there and start at that AUG. Okay, again, it's all chemistry that drives this process. Complementary base pairing, from the subunit to the Shine-Delgarno sequence, and then tRNAs to anticodons to codons. Okay, so that sets it up. So small guy hops on, tRNA holding methionine, right, because it's complementary at its anticodon, sits, that pulls in the large ribosomal subunit, and that would be initiation, okay, of translation. Again, just like replication and just like transcription, in translation, that's pretty much the only place that eukaryotes are different significantly from procs, right? Remember in replication initiation, the difference was one origin of replication for the prokaryotic circular DNA, a whole crap load of origin of replication, but otherwise replication was the same, right? Factors, open the bubble, primers, extend, blah, 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 right? Same idea, only initiation different. Transcription, Prox, sigma factor, binds -10, -35, brings in RNA polymerase. Ukes, TATA binding protein, binds the TATA box, brings in RNA polymerase and all the friends. Everything else is essentially the same, right? Same with here. In Ukes, this guy just grabs the cap. Instead of grabbing at the Scheindel-Garneau sequence, the small grabs onto the cap of the mRNA and slides to the first AUG. Complementary base pairing sets it up. Once we're here, everything else is essentially the same. Okay, so it's really just the setup of initiation difference between Prox and Ukes for translation. Okay, yeah. No, exactly. So this shows us that when this small guy sets up here, right, the AUG is in the P site. Yeah, so it doesn't start in the A site, it starts in the P site, or so they claim, and we're just going to believe them. And that leaves A open for the second one to sit there. So yeah, so this would be the end of initiation, or so forth. Once that's all set up, small, tRNA, large. And then, of course, you know, it has help doing all of this, but we're just doing the basics. Okay. Mm-hmm. They make a big deal out of this, and that is a difference between Prox and Ukes, is for whatever reason, the first AUG in a prokaryotic mRNA has the amino acid methionine is modified with this formal group, which I'd have to look up. I can't remember what the chemistry is. Remember when I said a methyl group is, you know, adding a CH3? Same idea. It's another little chemical group onto that amino acid. I don't think anybody really knows, and if they do, we don't care, the significance of that in Prox. If there happens to be another AUG someplace in the middle, it just gets a regular methionine in prokaryotes. So why, and how does it know? We don't know. Okay. It's got something, but I don't know why, and how does it know that that's the first AUG versus, unless it's just because it's in the P site and there's no large sitting there? I don't know. Like, that can't get in if it's already got the large guy sitting there, right? Right. In eukaryotes, it's just plain old methionine. It's plain old methionine if it's the first AUG, the second time you find one, the third time, the fourth time, it's just plain old methionine. So whatever importance it has in Prox, we've lost it in Ukes. So, I'm not going to get crazy about that, right, but in case that comes up, right, that's just one, and that's part of initiation, so that's, again, a difference between Prox and Ukes. So all in initiation. Okay, that's just reading the same things we already talked about in case we forgot to talk about it. Okay, so that's the end of initiation. Everything else is the same between Prox and Ukes. Again, that makes it easier. And elongation just keeps happening until a stop codon falls into or is read up to the A site. So if we continue on from where we were, zoom in a little bit. Right, after initiation, right, we have this guy sitting here. Here's our reading frame, AUG. The next guy is in the A site, right? This is the A over here. The acceptor site. Whatever amino acid has an anticodon complementary to this codon can dock there by hydrogen bonding, right? And then the catalytic activity of the ribosome can break a bond, make a bond. Okay. If a tRNA tried to come in here that was CCC with the anticodon, not going to fit, right? Won't sit there. Boom, gets kicked out. Okay, so only the guy that can actually sit and hydrogen bond in the A site. So this is showing, there he goes, he sat there. They're already showing us, if we really do an extreme close-up, whoa, holy crap. Right, this bond here between tRNA and amino acids already broken, right? So this is the one where I drew the red X. Break a bond, and then this one is they're showing a peptide bond, right? That's the classic peptide bond. Yes?
What kind of bond is between the P and the
Oh, somebody asked me that, and I don't know the answer. I can look it up, but I don't know what it's called. I know when you I know the process of putting the amino acid back on it, but I can't remember what that. That's by peptidal transferase, but we don't care about that. It's very tasty. So yeah, I don't know. But we break it, so we don't really care about it, right? We're not going to care about it till maybe 380. But I can look it up, because yeah, I should know, but I don't know. So but a good point is that, see, it's still intact in the one in the A site, right? The amino acid is still attached to the tRNA that's sitting in the A site. We broke it, the one that was originally in the P site, and now we've connected the two amino acids, and they're connected to the A site. So if we went and, right, this guy would be like a circle amino acid connected to a circle, and it's physically still connected to that tRNA. Okay? So when the ribosome shifts one codon over, right, it's going this way towards the 3' end, the whole thing will shift. So now the A site will be here, this will be the P site, this will be E. Okay? So when that happens, this guy is now naked, right? He's got nothing. He's got no amino acid. He's now in the E site and he just falls out. A is now in P and A is open, right? So this would be the new A, it's open. The next tRNA that's complementary to CGC, so it has to be GCG. That one will sit there, hydrogen bonding happens to dock it, break a bond, make a bond. Shift. Fall out, break a bond, make a bond, shift. Fall out, new one comes in, break a bond, make a bond, shift, all the way through. Until we hit a stop codon. So then they're just showing you this is after the shift, right? So this one went to here, so this would be the shift. Now A site's empty, right? It's ready for its complement to this guy. This guy's falling out. He's just a naked tRNA now, right? And the amino acids are peptide bonded together and still stuck in the P. That make sense? So another snapshot in time. A site's ready to accept another one. And so it does. Right, here comes this new guy. Oh, that's the same, sorry. In case I forgot to. Come on. Hello. Where are you? Aha! Right, so this guy's falling out, these guys are still attached, this guy is attached here, it's coming in, break a bond here, make a bond here, it's still attached to this A, 1, 2, 3 amino acids, remember this is the N-terminus sticking out there, and then this guy falls out. Right, and then elongation continues, the next 3, another one, break a bond, make a bond, shift. Okay. And then this is just to remind us that while this is all happening, as soon as the ribosome is away from the Shindell-Garno sequence and off of that first AUG, and there's room for another one, another small subunit just can grab the Shindell-Garno sequence. Another tRNA with a formulated methionine sits, large ribosomal subunit sits, acceptor site, another guy comes in, break a bond, make a bond, shift, and so you have, on one, this is your transcript, so what does that mean? It's mRNA, right? And we have tons of ribosomes, each making polypeptides. Right, so this would be a ribosome that's fallen off. That was the first one that docked. This is the one that just hopped on. Okay, and then they're showing us, right, the length of these, and then there should be free polypeptides if some already fell off, right? And they're showing us down here the length of these proteins is much longer than here. These guys just started. Maybe that's 5 amino acids and this one's 300. Right? So as you grow farther, the longer these guys are means the longer the ribosome's been on there. And then you'd automatically know this has to be the 5' end and this has to be the 3' end because it's traveling 5' to 3'. Yeah. Each ribosome goes through once and then falls off here, but then this guy could technically come back and bind and go again, or its parts. You know, the small and large fall apart, but yeah, they can be used again to retranslate that exact same mRNA. And so lots and lots of ribosomes at any given time. So again, it's not wait at the slide till the kid gets to the end and then you go, you know, at the water park. You just jump on as soon as that kid's out of your way. You jump on like you're trying to catch up with them and bash them, you know, in the head in the pool at the bottom. So to get as many kids down that slide as fast as possible rather than being safe and waiting until they get to the very end. Right, so we have all these guys. Right, we talked about transcription, the same thing happens. Right, the sigma can bind, an RNA polymerase comes in and starts transcribing. As soon as that guy's away from there, another sigma, another RNA polymerase follows right behind it. And then off of each one of those transcripts, there's a whole crap load of ribosomes. Right, so you can get lots of gene expression in a much shorter period of time than if you had to wait. Okay, that polymerase has to get to the end and fall off, then walk around, come back, take another turn. That ribosome has to wait till the end, fall off, come back, take a turn. Right? We're just transcribing the crap out of it and translating the crap out of each one we've transcribed. Yeah.
I don't remember if you mentioned it or not, but in use we have a control that we draw an RNA where we have a tail and a cap.
So you're talking about control of how long something turns over. As far as I know, no. So they still do get chewed up, so the turnover, but they can't control, they all get turned over about the same speed. As far as I know, there's not another mechanism for controlling that, right? Because they do have those endonucleases. It's just that, I was talking about this in my office. Okay, hang on. When we think about prokes, right, they're in one cell. Here's our euc, here's our nucleus, right? Everything's all together. And so if, here's their chromosome, they're doing transcription here, right? Technically, there's nothing protecting the end. There's no cap, right? This 5' UTR is just a regular phosphodiester bond. That could immediately get chewed away by nucleases. But immediately upon transcription, ribosomes hop on it. So it's kind of protected because there's a giant structure in its way. So a nuclease can't get at that DNA if the small subunits got it with the large, and then as soon as it gets any bigger, right, it's just covered with ribosomes immediately. So there's some protection, right? And then as it goes, yeah, it's probably going to get, and as soon as it chews through the Shine-Dalgarno, boom, translation stops. So it's not like, so they do have turnover of mRNA. Not every single RNA they make lasts forever and they're just stuck making it, right? Because otherwise the lac operon, you'd have all those stupid enzymes when you don't need them. So that would be most likely, right? And same with the 3' end. Once it does come off, it'll start chewing that way, and at some point it explodes it. In eucs, right, we have and we're doing transcription, but we can't have a ribosome hop on it because our ribosomes are out here. So by necessity, if we want it to survive, right, we have to 5' cap it to protect it because it's free to be chewed up while we finish that whole piece of transcription. We got to splice it during all that time it's available for nucleases, right? And then we got to tail it. So during all that time, we would have gotten nothing, and then, right, no survival. Okay, so let's finish translation. Again, right, we have this open site. We keep going, elongate until we get a stop codon in the A site, in frame, right, in that triplet. Again, if the UGA overlaps two different codons, it doesn't matter. It's whatever sits right here in the A site is what determines which tRNA sits there based on its anticodon. So it has to be in that reading frame to be a stop. You can have UAG all over the place in your mRNA as long as it's not in a codon. If it's just a triplet, but it overlaps two codons, it doesn't matter, right? It has to be in that site. As soon as that happens, this is a protein. It is not a special tRNA. Watch me write it down, which means you guys should be writing it. Not a modified tRNA, and no bullshit about any tRNAs. Okay? It's a protein that binds in this site and causes explosions. I don't know. Blows the whole thing apart. Right? It's like the termination and transcription mechanism, the big boot, right? It grabs that stupid hairpin and then kicks the crap out of RNA polymerase. Same idea. Release factor kind of looks like a foot. Kicks this guy right in the crack, blows off the large subunit. I don't know. in the video, it had little feet. Remember? This guy had little feet. It was like this. He had little toes. Right there. Yeah. In our video that we watched last week, it looked like a foot. No. But we're going to pretend like it looks like a foot so they can remember that it kicks its ass. Okay, it's a churd. Okay. And so, right, at that point, we still have some action here. We break this bond. And that releases our peptide, that releases our protein, and then we get a better view, right? See how it kicks its ass right there? And everything falls to shit. I had to swear because Dr. Stein's here. Okay. Normally I would say crap. Poop. Feces. Excrement. Doo-doo. Dookie. I'm a walking thesaurus. Okay. That makes sense? tRNAs go away, they're naked, falls to shit. Next one comes through. That's termination. Okay, no special tRNA. Oh my God. All right, Dr. Stein, we're not going to talk about this. I know. We already talked about this. We can modify proteins later, but we're not going to talk about them. That's one of the levels of gene regulation we talked about last week that's used, but the most important one, the mostly used, is the level of transcription. We're going to... I know. Okay. I know, but I like, who likes mutations? Anybody? But they're easy memorized questions for the exam. That's the only problem. Do you want to do this, or do you want to ask questions to the two of us? Do this quick? Yeah, it's super easy, right? Mutation. Ooh, something bad happens and you either don't complementary base pair correctly, right? Does this look good? Something wrong happens. It's not supposed to happen. Well, but it's not complementary base pairing. So that's wrong because everything is driven by complementary base pairing. Don't fucking argue with me. Just kidding. Right, it's not wrong, it's not bad. Mutations drive evolution. Right? The only way you get new alleles is by a mutation. Right? It's a mistake because it's not, right? A T is not supposed to bind to G. It's supposed to be a C. Right? But it doesn't mean it's bad. Mutations don't always cause something bad. They might change an allele that for a pigment and now you're brighter red and chicks dig you. So you get more sex, have more babies, pass on the bright red cheek allele, so your offspring take over the world. Okay. Wild type is the normal non-mutated mutant, right? Is the one that's a mismatch. And so then when replication goes on, right, the next round, if the template strand had a T, even if it was wrong, not supposed to be there, right, that DNA polymerase doesn't know it's not supposed to be a T. There's no knowing or thinking. It just chemistry, right, adds an N. And so then this strand would be mutant. The original strand, when it's replicated, would stay wild type. That could potentially create a new allele, could do nothing. Okay. No, yeah. Right. It's just passing those through the population for survival of the species, or of that population. Right. So don't put your testicles outside the little gown when they give you the lead thing for the x-ray. Don't, like, suddenly hop up and spread them in the dental chair, okay? Legs get under the lead. Okay. A point mutation is anything that's a single base pair change, a single nucleotide. Okay, it could be technically a deletion, a point deletion, point insertion. Usually they're talking about a change from an A to a T, G to T, G to C, some change in one nucleotide to another. An example of where one point mutation, one change in a base pair, results in one change in an amino acid in a protein that causes badness. Right? Lots of times, what? Yeah, my favorite protein. Lots of times, one single base pair change doesn't do squat. Right? It might not even change the amino acid based on the redundancy of the genetic code. Right? Lysine is this. Remember, wasn't that one the slut? Six different codons. Right? If you have a mutation that changes from one lysine codon to another lysine codon, it doesn't change your protein at all. That's a silent mutation. It's a mutation at the DNA level, but not at the amino acid level. If you change it at the DNA level and it's changed to a different amino acid at the protein level, then we call it a missense mutation. Sometimes that doesn't matter. If it's an amino acid that was basic and now the new amino acid is also basic and it doesn't change conformation of the protein, if two proteins with a different primary sequence have the same conformation, does that change its function? No, it's all about the conformation of a protein for function. Okay? So it has to change its overall conformation and either ruin the active site if it's an enzyme, or it reflects light differently if it's a pigment in order for you to see a phenotypic change. Okay? In the case of sickle cell anemia with hemoglobin, it's a one base pair change at the DNA level, a one amino acid change in the protein level, yet it totally screws that protein so that it now polymerizes for its quaternary structure differently than it's supposed to, which makes long extended fibers, which actually stretches out red blood cells. Red blood cells are essentially a sac of hemoglobin, right? And so that causes them to be pointy. They get stuck in your capillaries, cause extreme pain. They don't transfer oxygen well, and people die. Okay, so bad. So that would be a missense mutation at the amino acid level. It would be a point mutation at the DNA level. And this is why silent mutations, right? It changes at the DNA level, but it, oh, there's no picture of that. But it does not change what amino acid is in the protein, right? So again, lysine to lysine, right? That's considered a silent mutation. And then this is our little table. This is what you guys should know. A silent mutation change in nucleotide does not change, right, the amino acid. So obviously, if there's no change in amino acid, there's no change in phenotype. Right? Exact same protein. A missense, change in both, change in DNA, change in amino acid. A nonsense is change in a nucleotide that results specifically in a stop codon. So amino acid to stop, to nothing. So that truncates the protein, whatever. And then a frame shift is if there's an insertion or a deletion, right? So instead of just changing one base pair to another, we remove one. And then when you do transcription and you make your RNA, that changes your reading frame. So every place past or with that missing or added nucleotide, right, changes your reading frame. So then every single amino acid past there is going to be different. So if you compare this guy down to this guy, right, this is where did they add one or did they take one out? They added one. This is where they inserted, right, that C, and from there on, all of these triplets are different and all these amino acids are different. So that totally screws your protein. Yeah. And so it's going to keep going until it just randomly runs into a stop. Not the original stop because it's out of frame. Yeah, it just then, right, it was added in the DNA when it's transcribed, right? It just ends up, and the frame, that's why they call it a frame shift. The frame literally shifts over, and so every single codon's off from that point. It'll eventually hit a stop or run off the end of the mRNA. But generally, yeah, there'll be some stop randomly in there. And that is it. Now it's time for questions, comments, concerns. For me or for Dr. Stein. Cumulative, not cumulative. Oh, I can't strike that, can I? Sorry, I'm totally ignoring you now. Yeah. And like some of the examples, when I say, remember back to chapter 4, we talked about the nucleotides, 3'OH, 5'phosphate. Yeah, all of that stuff, peptide bond, just came back to haunt us, N-terminus, C-terminus. Yeah, I never talked about that again, so. Yeah. $10,000 points on this exam. Are we going to have a paper? You're not going to have something on paper? Yeah. Paper gives paper cuts, and it's not okay. But you'll probably have fill in the blank, similar to like your regular. And I won't have any cumulative drawing stuff. So the 50 points of the final will all be, and I don't know what I'm doing for the other stuff for tomorrow, because I didn't do anything this weekend. No, so we're not going to draw that again. But you might see drawings of those in the cumulative part and name this phase or tell me if it's G2 or how many chromosomes or, right, that kind of stuff is fair game for the cumulative part. But not you drawing it. You'll be given my drawings or book drawings or whatever to identify. Yeah. Yeah, it's going to all be mixed together. That's really the only way to do it through here, unless we had two separate exams and you upload one, submit one and then start the other one. And we figured that's a lot of chaos for, in this 2 hours. So yeah, they'll be all mixed up in there. I mean, some will be obvious which ones are. Okay, she's going to ask about delta G, so you guys better go back and look at that, because she already talked about it. You know how much she loves that. That and vesicles. She's going to mix up a vesicle and delta G question. And then she'll be like frothing and, just like me, just wait till I talk about phosphodiester bonds and comparing replication, transcription, and translation. I'm like getting excited about it already. So, So, those are going to happen to you for me, and so you'll be able to go, oh, this is a Stein question, it's about this, or this is, it seems pretty compartmentalized to glycolysis. Like, we never talked about glycolysis again, except when we were talking about crapping your pants and lactose intolerance, right? Yeah, the section of this new stuff is going to be like a standard, I need you to understand it. They're not trick questions when I change something. I want to know if you know the process and can think through it, you can accept a change and figure out how to do that. So that way, we're assessing critical thinking skills, because that's really what science is. You have to know the language and the vocabulary, so you have to memorize stuff, but science is figuring things out. That's the great part and the hard part about it. Yeah. So generally, the inducer itself, I'm lying. For your examples, the inducer itself generally binds to something else that contacts the DNA, right? So the inducer, we call it that because its presence in this cell causes gene expression, right? So for us, having lactose inside the cell causes, right, here's the repressor, right? If this guy grabs on it, its presence is there, that guy has to come off. Right? And we had sigma and RNA polymerase sitting there waiting. Now he can go and do transcription. When the repressor was sitting there, nothing can happen. Once lactose gets in, it induces the expression of the gene, not by positive regulation. Positive regulation of an inducer would be that inducer itself goes and binds to him or something, and makes him go, or something binding and go. In this case, you can think of it as pulling off the repressor and allowing polymerase to do its job. Right, and so then these guys for the lac operon are all the genes that are then transcribed into one big thing, right? And then they're translated into 4 different proteins. And so these guys are usually all in some sort of biochemical pathway, right? All of them are needed to break down lactose, or all are needed to synthesize this tryptophan. So you either need all of them or you don't. And so they simplified it by they're all on or they're all off. Beta-galactosidase, yeah, and actually, yes, yep. So they're coordinatedly regulated, because you either need all of them or none. Whereas in eukaryotes, there's very few proteins that always in every single cell are needed together or not. Right? This cell might need 2 of the 4, but this cell needs 2 others of the 4. It would be a waste of energy to turn on all 4 if we only need 2 of them. So in eucs, it's promoter gene, promoter gene, promoter gene. We don't make a big RNA and then translate into 4 proteins. Yeah. Hmm? Yes. That would be like if you have a translocation. So in the immune system, if you get strep throat, your B cells are just jamming out antibody molecules to try to get rid of that bacteria. Like you just jam on transcription translation a shitload. If you have a problem and you have a translocation where another gene gets stuck in front of that promoter regulatory unit, you just start making a shitload of that protein. And so many leukemias and lymphomas are a translocation of an oncogene, a cancer-causing gene which makes the cell cycle go, c-Myc, BCR-able, all kinds of things. And so now we're making a crap load of this protein that normally is fine. Normally it says, go when you need to do mitosis, but now you're making 1,000-fold that you're supposed to, and and then you get cancer, right? That cell keeps going, allowing more mutations through each time because replication's happening way too fast. They're not catching anything. The repair mechanisms aren't working. More and more mutations, faster and faster and faster, and then you get full-blown cancer. So yeah, that definitely happens. Yeah, so gene regulation's super important, right? They need to be on when they're supposed to and off when they're supposed to, at the right level as well, or, right, all hell breaks loose. Okay, what else? Glycolysis, photosynthesis, delta G, delta S. He does like delta S too. What else? They can go if they want. They insisted I save time for this. Remember, you guys all wanted a whole hour of this. Do a lecture capture so I can get all these questions. And I said, you know what? I know what they're going to do. They're going to sit there and stare at me. So I'm not going to take time out of my weekend to work for free to do a lecture capture so we could stare at each other. Much as I love looking at you guys. Yes, ma'am. Thank you. That's a stunning question. Yes, please. I missed out on that. Ooh, I love scurvy. That's why pirates have no teeth. They got no fresh fruit or vegetables on a boat. They eat pickles and pickled eggs and beef jerky. Anyone? Bueller?